While trained at it is initially working on DNA repair and recombination gene at Arabidopsis thaliana. Over the last, François lab has been interesting in using to develop novel approach in plant breeding, mainly in soybean and barley. Aim to act as a bridge between the lab-based genomic science and there are numerous applications in the and improve crop varieties. Working closely with industry partners to join and develop and implement genomic infrared breeding. Is professor at the University Laval, Canada, and so we will let him the place his talk of today. Thank you.
Thank you, Sébastien, for that introduction. Let me start the sharing process, and hopefully you can confirm that you see my full screen. Okay, so as Sébastien mentioned, I'll be talking and sharing some of the work we've done over the last decade in developing some genotyping solutions and tools to assist us in basically the germplasm characterization, breeding in barley and also in soybean, as you'll see. As he mentioned, I work in the School of Agriculture and Food at Laval University, and I'm also part of an institute for integrative and systems biology, the IBIS, that you'll see a few times in the talk. And so basically, I wanted to start by giving some historical perspective on the development or my own personal history of work with DNA markers. So when I started graduate work in the mid-80s, we had very few options, and basically this was RFLPs. And as the PCR technology was developed in the late 80s and really took off in the 90s, then a list of PCR-based technologies appeared, such as the rapid markers, SSRs, and AFLPs. And fortunately, later, maybe 10, 15 years ago, SNPs really came into their own as the main genotyping technology in crop systems. And throughout my journey, this use of DNA markers has been used in various types of research contexts. As a graduate student and postdoc, I was mostly involved in studying plant transposable element systems in tomato and Arabidopsis. And then, as Sébastien mentioned, I briefly, for about a decade, was interested in working on DNA repair and recombination genes in essentially in Arabidopsis and a bit in soybeans. And since the last maybe 15, 20 years, most of the work has been more related to crop genomics and working on barley and soybean, doing some genetic mapping, doing marker development, GWAS, and all of these other aspects that come with this type of work. And so just for the students and postdocs that are too young to remember these old days, this is what work used to look like with RFLPs, and it was, I can assure you, an absolute nightmare compared to what it is today in terms of working with radioactivity and taking so much time to develop markers. You know, doing a single marker assay could take a week or two just to get the information. So it's a very, very painful process, and really crawling along and building a genetic map could take anywhere from 6 months to a year. So it's very, very laborious. And then PCR came around, and now we could have marker systems where you could interpret or analyze multiple loci on a single gel. And so that was definitely a step forward in terms of the speed with which we could perform our assays, but it was still a very limited degree of parallelization in terms of how many markers you could analyze at a time. And then fortunately, then the SNPs came along, and they offered then a much higher, much degree of parallelization with thousands and sometimes many thousands of markers that could be assayed jointly. And so that was a really big step forward. And I think it's had a really big impact on research as experienced by those who practice it in the lab. And so when I think back to what life was like as a graduate student or postdoc, we used to spend a tremendous amount of time in dedicated to data production, and that left very little time for data analysis in proportion. So we spent a lot of time at the lab bench trying to laboriously generate all this data. And I think nowadays we have a much more promising and interesting scenario where now data production itself is a much smaller effort for those involved at the lab bench level, and it allows a lot more time to perform data analysis. And so I think it's had a really major shift in terms of what it means to be doing, for example, molecular genetics and genomics. And I think it's a very exciting time to be doing this because we really have access to an abundance of data, and usually we don't have to wait ages to get the data with us. And that gives us much more time to be asking meaningful questions that in the realm of biology. At the same time, it's also challenging because I think that when I think back to what was required of me, I was, in a way, as a graduate student, a high-level technician in the sense that a lot of the skills were related to, generating the data. And I think today's students are confronted with much more high-level conceptual analysis. When I have to explain concepts like LD, these are not run-of-the-mill concepts. It takes some while to get accustomed to them and to master them profoundly. So I think it's to me, this is a very positive development, but it has changed quite dramatically what it means to be doing research in this area. And so what I wanted to go over today was to basically have a discussion on the field of genotyping and what it can mean. So there are obviously various scenarios where you might want to be doing genotyping. And in barley, at least, this runs a very wide range in terms of the scale of the genotyping. So if you're in doing breeding and interested in conducting marker-assisted selection, you may have one or a few markers that for which you're interested in conducting marker-assisted selection. And then as you move along, then you can require more markers to do more sophisticated things or broader things, fingerprinting cultivars, for example, for regulatory reasons, can require a few dozen markers. You can look at genetic mapping, which typically would require, maybe a few hundred to 1,000. You move into GWAS, genome-wide haplotyping, and ultimately, when you reach the reference genome scale, you're looking at billions and billions of sequence information that's required. And so fortunately, there's a whole range of technologies that we have access to today that allow us to meet many of these needs, whether at the low end it be CASP assays for assaying single markers, or whole genome sequencing and genome assembly at the more exhaustive level. And for much of what we conduct on a daily basis, we're more in the medium density situation where tens of thousands, 100,000 SNPs is quite sufficient to conduct many of the types of analyses that are very common. And for that, then there are tools such as SNP arrays, and at the higher level, people can be using skim sequencing to harvest many hundreds, thousands, or millions of SNPs. And on our end, we've been using mostly complexity reduction methods that have different names that relate to very similar things, whether it be GBS, RADseq, the ecology field, as you tended to call this, RADseq, and in China they also have what's called SLAFseq. And so basically they're very similar technologies that have been used to yield a number of markers within that those different areas. And what we've tried to look at is basically trying to see if we could expand the realm of GBS to meet some needs that went beyond what was the initial range of markers. So either at the high end for reaching into the numbers that typically would be obtained through SkimSeq, even at the low end, sometimes this can be useful, and even trying to incorporate single marker assays in the context of breeding efforts. And so I'll try to run through these different applications and show you how we've developed tailored or customized solutions to try to address these needs. And so in terms of defining one's needs, I think it's a really important step to start with, and to ask oneself, well, how many markers do I need to conduct this or that type of work? Do I need to have only anonymous sort of genome-wide markers, or do I also need to include trait or gene-specific markers in the breeding realm? What's an acceptable cost given the scale on which these analyses are going to be conducted? If you're only analyzing a few hundred lines per year, you're less susceptible to being constrained by cost. But if you're doing things at a very high level in the tens of thousands and above, then cost becomes quickly a very important issue. In what time frame can these analyses be conducted? Do you want a fixed set of markers, or are you flexible in terms of having a slightly different catalogs of markers coming out? And in terms of the analytical side of things, do you want a turnkey solution where you simply get the genotype data, or are you in a position to conduct some of the bioinformatic analysis? So these are all questions that one is confronted with when adopting a solution for meeting those needs. And so in what I'll be talking today are variations, basically. So I call these the many flavors of GBS. And so I'll start by quickly going over what is our standard GBS protocol, and then walk you through some of the variations that have allowed us to develop variant versions of GBS that can either allow us to go at a higher density, get more markers for some applications. In other cases, we're actually interested in getting fewer markers, but with the benefit of reducing the per sample cost. And in another scenario, we're also interested in adding onto the genome-wide markers, markers that are already in use by breeders for trait-specific marker-assisted selection. And finally, I'll close with a very, very technical aspect, which has to do with basically what we call nano GBS, which is a miniaturized version in terms of reaction volume, again, aimed at reducing per sample cost. And so to start, I'll have a brief run through of what the GBS technology relies on. And so basically it's a sampling process through which we want to interrogate a small portion of the genome and to examine it for the presence of SNPs. And so in this particular case, these complexity reduction methods basically use restriction enzymes as a point of entry into the genome, and whereby once these cuts are made, we will be sequencing DNA on either side of the cut. And fortunately, when you look at a collection of samples, most samples will share common restriction sites. And so we will be interrogating the DNA sequence flanking these sites in homologous context. And so when we do find the presence of a single nucleotide polymorphism, then that informs us on the presence of a marker in that area. Now, that's the basic concept. In terms of the sort of a lab methodology, well, basically it's a fairly simple process where genomic DNA is digested with restriction enzymes, and then we add through ligation adapters that have both a barcode to identify the sample from which a particular restriction fragment was derived, and also some primer sequences for the purpose of next generation sequencing. And so it's a fairly simple process where samples are laid out in 96 or 384 well plates, whatever design you're using. Digestion ligation occurs individually in the sample wells, and once everything is barcoded, then everything can be pulled together into a single library that undergoes various steps, including quality control and eventually sequencing and bioinformatic analysis. So for us, the standard or workhorse protocol for barley is based on the protocol developed by Jesse Poland's group and published in 2012, which typically will, for us, essentially yield in the area of 30 to 50,000 SNPs for a diverse set of accessions. And within the context of a biparental cross, if you're doing QTL mapping, for example, then you're looking at progeny of just 2 lines. You'll have just a smaller number of markers, obviously being polymorphic in that restricted setting. And oftentimes these are performed on a 96-plex scale. And for us internally, the cost is about 10 to 15 euros, pounds, or American dollars. They're about, I think, not too far from parity at this time. So that gives a good indication of the cost, which obviously has many considerations depending on the scale of the analysis and your status as a client. But that's really a very affordable method for us to perform genetic analysis. And then once we have these marker sets, then we're obviously interested in using these to perform biological experiments, such as in this example, performing GWAS, in this case, showing results for measuring days to heading in a population with a fairly good sized catalog of markers. In this case, we had 44,000. You can conduct single SNP analysis as shown on the left, but you can also explore alternative approaches where, for example, you will first define haplotypes with these SNP catalogs and then look for associations between haplotypes and your trait and see if there are novel things that pop up in doing things this way. And so that's the very standard GBS protocol, I think, that most of you are highly familiar with. And in the course of our work, certainly we've found the need to develop variant technologies that would address some of these needs. So I already indicated one being an increased number of SNP markers, and I'll also show an example where we wanted actually decreased number of markers. So in the first example I'll be going over is what we call high density GBS, and as the name implies, it's really to get more information. And I think as a backdrop to understanding how one goes about trying to develop alternative protocols, you have to have an understanding of what it means to customize GBS, and I like to refer to this as finding the sweet spot. So when you're digesting genomic DNA, you have obviously a choice of enzymes to consider, and depending if your enzyme cuts at a high frequency, a medium frequency, a low frequency, it will essentially result in your genome being fragmented into a very high number of restriction fragments or a low number of restriction fragments. And then when you proceed to sequencing, you will usually allocate a given sequencing effort or sequencing budget to doing this characterization. And if the amount of sequencing that you carry out is limited, well, in the example shown on top where you have a high frequency of cutting, what you'll notice is that many of the restriction fragments will have no coverage by reads simply because you just have too many fragments and not enough sequencing reads to be able to characterize them. And so that's not a very fruitful way of doing things. You'll have a lot of missing data and won't be capturing as many SNPs as you might want. In the bottom scenario, we have the reverse situation where basically you have too few restriction fragments and you're reaching excessive coverage of those particular restriction fragments. And so the sweet spot in this scenario is illustrated in the middle, where basically you have an adequate number of restriction fragments to ensure that they will each enjoy coverage that's sufficient to perform a genotype call. Now, in the example I'm showing, obviously, we're thinking about a diploid species with limited complexity where you don't need to have a high number of read coverage to be able to call a genotype, for example, if you're looking essentially for homozygotes. So basically, again, the idea is that if you don't have the right choice of restriction enzymes, you can spread your sequencing effort too thinly, and that will not be very good. And the ideal situation is where you have obviously adequate coverage for most of your restriction sites and don't venture into a situation where you have an excessive concentration of your reads onto a small number of fragments. And so basically in trying to develop what we call our high density GBS, that was the crux of the matter, was trying to identify a combination of restriction enzymes that would meet that particular need. And so to help us in doing this, we had to define what it is that we were aiming for. And in this particular technology, we had actually developed it for soybeans. And in this particular case, we were aiming to get over 500,000 markers in a diversity panel, where typically we would get more on the order of 50 to 100,000 using a standard GBS protocol. And that was really to allow us to get what we call a uniform coverage of the physical map. Uniform being understood in the context, not necessarily perfectly uniform, but certainly within the euchromatic regions, we were striving for a highly uniform coverage. And this would be used for doing high resolution GWAS studies or genome-wide haplotyping. And so essentially we're aiming for a cost that would be in the 50 to 20 euro pound realm, and alternative technologies to conduct a similar type of analysis would have been either large SNP arrays or skim sequencing. And this would be for applications of low to medium scale, where we're looking at tens or hundreds of samples for these types of analyses. And so in trying to devise a protocol of this type, we first looked at or considered different restriction enzymes and how they would essentially digest our soybean genome in this case. And so we used a tool that we developed called Depth Finder, which has the ability to basically to simulate in silico digestion with different enzymes and to report on the distribution, the size distribution of these different fragments. And in trying to think about the targets we have, we thought that we needed to aim for maybe 1 million SNPs and see how to work from there. And what you can see from the graph shown below, so it's a size distribution. So on the y-axis, you have the number of fragments that fall within a given size window. And on the x-axis, you have these different 100 base pair bins, ranging from, on the left, 100 base pair to, let's say, 100 to 199, then 200 to 299, so on and so forth, up to the 800 base pair window. And what we found by doing these simulated cuts was that some of the enzymes or enzyme combinations simply yielded too few restriction fragments. So there was very little chance that we would be able to get the number of informative SNPs given the limited number of fragments that would be in an appropriate size window for being captured through GBS and sequencing. In other cases, there were just too many, and they tended to be very, very small fragments, so in the 100 base pair window size, and that's not going to give you a very broad opportunity to capture SNPs. And so in the end, it turned out that the BFA1 combination or enzyme, choice of enzyme, was the one that seemed to best suit our needs, with an estimated number of about 1.5 million restriction fragments and with a better size distribution. So we opted to use this enzyme to develop the high density GBS protocol for soybean. And what you can see in this particular graph is the results in terms of the number of SNPs that were obtained and comparing it to what would be obtained in the same panel. So it's one set of 96 samples that were analyzed through SkimSeq, and then the same samples were analyzed through standard GBS, a SNP 50K array, and then the high density GBS that I've just described. And as you can see on the, as far as the number of SNPs are concerned, well, we're within the window we'd hoped for, with close to 900,000 SNPs being uncovered in this particular instance of, or for this panel. Now, looking at SkimSeq, you can obviously get more markers through this technology, depending on how deep a skim sequencing you perform. So at the 1x level, you can reach very high levels of SNPs, but that requires a very significant sequencing effort. And you can see that the standard or more routine technologies of standard GBS and the SNP, the soybean 50K array, yielded much fewer SNPs in this panel. Now, in terms of the missing data, you won't be surprised to learn that with SkimSeq, because it's a very shallow sequencing technology, that yields a lot of missing data, whereas with the high-density GBS, we had much fewer missing data points, and so that was obviously a strong point. And in terms of cost, there was quite a substantial difference between the high-density GBS and the skim sequencing technologies that could yield comparable numbers of SNPs. And so for all these reasons, this could made for a nice alternative to skim sequencing for us when we were interested in collecting very large numbers of SNPs using a GBS approach. And so the key elements here was to do some preliminary research into finding an enzyme that would produce a sufficient number of restriction fragments and with an adequate size distribution. Obviously, once you have that, then you can adjust the scale of sequencing to ensure that you have sufficient read coverage to be able to make good calls on your genotypes. And a note that I'll come back to later is the fact that this whole technology can also be, the price of the technology can be reduced by conducting what I'll illustrate later as being a miniaturization. So that's at the high end of things. But in other cases, we've been interested in doing what we'll call low density GBS. And in a lot of work that's done in breeding, for example, in building genetic maps, in doing genomic selection, you really don't need very high numbers of markers to conduct that type of work. And when we tried to define what the needs were for this particular technology, we were looking at getting 4,000 to 6,000 markers among a diverse panel of lines, but that's just as a reference point, because the most common use case is to be looking at 1,000 to 2,000 markers between any pair of parental lines. So whether you're doing genomic selection within the progeny of a cross, or whether you're trying to build a genetic map for a given, for do some QTL mapping within a biparental set of progeny, 1,000 to 2,000 markers would be sufficient to achieve both of those goals. And so you're obviously aiming to have a fairly uniform coverage of the physical, but most importantly, the genetic map. And because these types of analyses, especially on the genomic selection side of things, can be conducted on a very large scale, cost really needs to be minimized to make this an attractive technology. And so in going forward, and this is recent work that's just been completed by a postdoc in the lab, we found that a 2-enzyme protocol involving a fairly rare cutting enzyme called the SBF1, along with a frequent cutting enzyme, MSP1, provided a really nice solution for this. And so here we'll just cover some of the major metrics of this low density GBS protocol. And I've indicated on the right hand of the table various results based on how much the allocation of a sequencing effort, ranging from 50,000 reads per sample all the way to 600,000 reads per sample, and how that affects these different metrics. First metric is the portion of the genome that we're capturing through this protocol, and this is called genome coverage. And so you see that we're capturing a very, very small portion of the barley genome, less than 1%, so from 0.2 to 0.4% basically. But this is enough basically to derive SNP catalogs that range between 3,000 and 7,000, which was within the realm of what we had aimed for in setting up this protocol. And even at these fairly low numbers of reads per sample, the mean depth per SNP was still fairly high, ranging between 10 and 85, and that results in a fairly small amount of missing data that in these scenarios are very easy to impute. Now, in terms of map coverage, the first we'll consider deals with the physical map, where you see that, again, across this range of sequencing coverage, you can express things as the number of SNPs per megabase, and we range from 0.6 to 1.3. But maybe more importantly, looking at the number of gaps that remain on the physical map, and you can see that gaps of 10 megabases, 20 megabases decline as you increase the sequencing effort. And looking at gaps over 20 megabases, we can basically abolish them completely. But even considering this, we have to recognize that most of these large residual gaps reside in pericentromeric regions that are relatively gene poor, that we typically do not put too much emphasis on when carrying out these analyses. And maybe more importantly than on the genetic map is to see how this number of markers can be distributed. And you see that we also can consider how many gaps of a given size remain on the genetic map. And you can see that large gaps of more than 10 centimorgans can be readily abolished by having a sufficient number of markers based on obviously the number of reads that you have per sample. And so considering the all different aspects of this, then we came to the conclusion that the 100,000 to 200,000 reads per sample, which is a very small number when you consider what you usually allocate to GBS genotyping, would be sufficient or provide a sweet spot, as I mentioned earlier, in terms of the number of markers we could obtain from that and the uniformity of coverage of both the physical and the genetic map. And so that became our protocol of choice for conducting this low density GBS, where we're after a reduction in cost and sufficient coverage for many breeding applications. Now, in discussing with our partners in the breeding realm, they were interested in having another aspect to this. They were very interested in having this sort of very cheap, low-level coverage, but they were also ideally interested in having information on trait-specific markers that they usually use in their efforts to conduct breeding for marker-assisted selection. And so we tried to see how we could add that component. And so basically, it's the same low-density protocol, but with the inclusion of additional SNP markers that are already in use by breeders. And obviously, when you're collecting or capturing 1,000, 2,000 markers in a random fashion like this, the odds of, by chance, stumbling on a marker that's already in use by a breeding program is very, very small. And so it has to be designed into the process. And so in this case, we decided to look for a series of markers that would be of broad interest to our breeders. So we asked them, well, tell us what markers you're using, and we complemented that with others. You won't be surprised to learn that many of the resistance or the markers that were suggested to us had to deal with disease resistance genes, or and we've added also some QTLs that were from the literature that seemed interesting. So morphological or phenological traits, photoperiod, vernalization requirements, and so forth, and also grain composition traits such as amylase contents or protein or starch synthase pathways. So those were the types of markers that got integrated into our panel of trait or gene-specific traits. And so these were already, for some of them, already in use. And we tested this implementation on a set of 16 barley accessions and included 5 of which had a reference genome available so we could check the quality of the data. And so what it boils down to is basically joining 2 different genotyping technologies, a low-density GBS on one hand and amplicon sequencing on the other. And so the trick for us was how to integrate those 2 to minimize the cost per sample. And so the approach is illustrated here. So on the left, we start by designing primers for these gene or trait-specific SNPs that are of interest to breeders. And so having primers being designed with a green portion which is basically the locus-specific portion of the primer, and then building in restriction sites into these into the extensions or the tails of these primers to ensure that we could capture these amplicons in our GWAS library production, and then a further extension of the tail sequence to ensure good digestion. So moving to the right side of the slide, we can perform multiplex PCR with these various sets of primers, and this results then in amplicons that house the various SNPs, A, B, C, and all the way up to 36. And then in building the GWAS library, we undergo the usual digestion ligation steps that you are familiar with, and these get captured through barcodes, barcoded adapters, and we'll move on eventually to sequencing. And then through bioinformatic deconvolution, we can basically assign reads as being derived from these amplicons that cover these specific SNPs. And by using this, then we're able then to output very clear sort of results files where we can define specific alleles at these genes of interest, and that's of interest or use to our breeding partners. And so just looking at some of the technical questions that this raises, so we have two essentially components entering into our GWAS library production. One is a set of PCR amplicons that are these 36 amplicons that I described earlier, and the other is undigested genomic DNA. And so we need to mix these two in some proportion so that the allocation of reads to both portions of the work is appropriate. And we can simulate this by looking at the concentration of PCR products and converting this into a number of copies of the molecules versus copies of genomic sequences. And we can simulate how different dilutions would result in a allocation of reads to the amplicon sequencing versus the GWAS portion of the work. And based on these simulations and empirical work, we found that a dilution of 10 to the minus 7 of the PCR products, when adding them to undigested DNA, would allocate approximately 10% of the reads to the amplicon sequencing portion of the work, which seemed to be a good fit. And so this is a slide in which I show a heat map illustrating the extent of coverage of our 36 amplicons among the set of 16 accessions. So the accessions here are the rows, the SNP loci or markers are the columns, and the color reflects the extent of depth of coverage of these amplicons. So it extends from black, which is failure basically, where we have fewer than 2 reads for a given amplicon within a given accession, and then a dark green being a very high level of coverage, so exceeding 99 or exceeding 100 reads, I should have added, as being success. And so you can see that depending on the dilution factor, it has an impact on the extent of coverage of your SNP loci. And you could look at this and say, well, why not use 10 to the minus 6? Because it results in better coverage of the different amplicons, but it does come at a cost, and this is the percent of reads that are allocated to amplicon sequencing versus GWAS. And what you can see is that using a less dilute PCR product will result in better coverage of the various amplicons, but at a very high price, meaning that half the reads are going to be allocated, in this case, to the amplicon sequencing, leaving only half of the GWAS portion, which is not ideal. And so this is, again, we're illustrating that the 10 to the minus 7 was a very good compromise between the adequacy of the coverage we could obtain for the amplicons resulting and keeping 90% of the reads for GWAS SNP calling. And so that's why essentially we have opted for this as being our optimal dilution, and it results basically in most of the amplicons being successfully amplified in all samples. It's likely that the 3 amplicons in which most of the missing data are concentrated simply have poorly or suboptimal primer design, that this could be improved. At the other end, it's probably not realistic to hope that any pair of primer will work on large sets of accessions and never fail. There are always going to be polymorphisms that lead to amplicon amplification failure. And the other aspect that was of interest to us was the data quality. And so again, because 5 of the barley accessions that we used in this work, we had assemblies, reference genome assemblies for them, we could simply ask, well, is the genotype that's called based on this amplicon sequencing, does it match the known genotype or nucleotide at that position in the reference genome? And over 99% of the genotype calls turned out to be accurate. And so that was satisfactory for this particular type of work. And in the final segment of my talk, I wanted to introduce what we call nano GBS, but which could be broadened to more than GBS. And so it's basically a technology to try to miniaturize the scale of our NGS library preparation. And this leads to reduced cost per sample, which is, again, when conducting large-scale work, is always very important. And that came out of a realization that when we were conducting our GBS or library preparation, we would end up with a 50 microliter volume, of which only 5 microliters were being used for pooling to assemble the final libraries, and thought, well, it's a bit wasteful to have these reactions done on a scale which is excessive in terms of what's needed for the final library construction. And so we turned to a device that's called an acoustic liquid handler. And so basically it's a technology that can allow liquid transfer at a very high precision of very small volumes of liquid. And so this particular machine works by basically emitting sound waves at the bottom of a well that contains a liquid, and that results in the ejection, if you will, of small droplets of a very precise volume, in this case 25 nanoliter droplets, that simply are ejected from a source plate, which is, let's say, in their normal orientation with openings on the top, and it ejects them to another well, 96 well or other type of plate, which is head down, basically. And so the liquid simply is ejected up into the empty well in this fashion. And you can see on the time-lapse photography on the bottom how this is happening in real time with these micro droplets being ejected out of the source well. And so in using this, we're able to basically conduct library construction, and again, illustrating this for GBS, but it's true also for any type of NGS library construction that we conduct. And so essentially we can go from, on the right hand, the standard process where we would normally use multi-channel pipetters and work with whatever, volumes allow us to get precise transfer of volumes, to move to the left-hand side with this acoustic transfer. And that allowed us then to reduce the scale of our libraries tenfold. And then that resulted in significant savings. And in terms of, are these things comparable in terms of output? So again, the technology was developed in the soybean realm with a set of 96 samples. And on the graph on the right, you can see the distribution of sizes of the fragments. And so the people who work with this call this the Bart Simpson hairdo. So it's got these spikes that reflect basically the distribution frequency of certain fragments in the population. And in blue, you have what the profile of fragments looks like with the standard protocol. And the red is the nano GBS. And you can see that they're very similar. And when you build and sequence libraries using these, you can see in the Venn diagram below that the catalogs of SNPs that were called using the same samples, but these two different GBS library preparation protocols, they overlapped almost perfectly. Out of 34,000 SNPs, 31,000 were common, and you see there are little edges on either end, which are unique SNPs that were called only in one protocol or the other. So very high degree of overlap between the two library preparation protocols, and the genotype calls were 100% identical when we had data for both, for one locus for both technologies. So it provided a very high degree of repeatability of the genotyping. But in terms of costs, then depending on the scale on which you conduct this, then it can lead to very significant reductions in costs. So we've broken these costs down into, let's say, reagents and consumables would be probably a more complete labor and sequencing. And by transitioning away from standard pipette-based liquid transfers into acoustic transfers, we can reduce the cost of reagents quite significantly. And in the end, we figure that we could achieve approximately a 2/3 savings in terms of the per sample cost, but quite significantly reduce plasticware by approximately 90%. And this became really important during the COVID years because, as you all know, there was a huge demand for PCR tests for COVID testing. And there was quite sometimes difficulty in obtaining pipette tips. And so by moving away from those tips, the timing was very good for us in terms of not being impacted by sometimes a shortage or delays in obtaining the tips. And so going back or reviewing the overall talk, you can see that across the range of needs that researchers might have in terms of scale on which they would like to collect data on SNPs. We've described today a range of GBS technologies or derivatives that can basically cover this broad realm of needs and provides useful and low-cost solutions for many of the activities that are conducted in my lab. And in closing, I want to recognize the work of the people that were the ones really doing the work. And so Amina was very pivotal in doing much of the barley work and involved in the design of the different amplicons for the AMP-seq version of the low-density product. Maxime is a postdoc in the lab that was working with developing lots of this combination of low-density GBS and the AMP-seq version. Dawood was a former student who's now a faculty member in our department and did a lot of the soybean development with the high-density GBS and the nano GBS work. We had lots of help from the folks in our genomic analysis platform, which is a service lab basically that can offer these services for a fee, and people in the bioinformatics that also helped us build pipelines to run through these analyses. And in doing much of this barley work, I've enjoyed the collaboration with breeding partners, Anna Badea and Raja Kanal in Agriculture Canada, as well as Aaron Beatty at the University of Saskatchewan, numerous funding partners for both the soybean and barley work, especially the Barley Council for much of this genotyping work that I just described. And with that, I'll end my presentation. We'll be happy to entertain any questions that you may have. So with that, I'll end here, and I'll probably turn off the screen sharing so we can go back to seeing people. There we go. Okay.
Thank you, Francois, for your presentation. That was really excellent.
Thank you. I'll be happy to take any questions.
So if anyone has a question, could they please put their hand up?
The time that people ask, people
Hello,
can
everyone.
write this. Okay.
My question is that I investigate the genetic diversity in Convolvulus arboris. It is a species of Convolvulus, a member of Convolvulus, in two different habitats by using ISSR markers. Are you hearing my voice clearly, sir?
Yes, yes.
So my question is that is it possible that can we use these GBS techniques? They may provide us the same result as in control or experimental site in different traits. Is it any kind of problem regarding this? The habitat may disturb this technique.
So a key consideration to start would be, do you have a reference genome for your species of interest?
Yeah, my species of interest is in Convolvulus arvensis, Arvensis.
Okay, so if you have a reference genome, then GBS technology certainly can be used. Even if you didn't have one, people in ecology also use approaches where they can analyze GBS-derived data and extract meaningful SNPs out of them. It's just much easier and I think much clearer when you can build on a reference genome. So all of the technologies that are used for GBS are applicable across a very broad range of species. I was mentioning that the genomic analysis platform, so the service lab at our university, they handle samples from hundreds of different species. And so it's a very, very broadly applicable technology. It does require, though, that you acquire some ability to perform bioinformatic analysis. There are pipelines that exist that can help people work through this, but certainly compared to the direct interrogation or examination of a gel, it requires a bit of skill, but the reward is that you will probably get markers on a much larger scale and ones that are probably easier to interpret than large sets of ISSRs for which their location is more limited. Their number and location requires more lab work to generate similar levels of coverage.
Yeah, thank you so much, sir. Thank you so much. Bundles of thanks for your answer, sir. Sir,
Any other question?
One more question. Sorry for that. Is there any open position for PhD under your supervision?
Not currently. I see Luke's hand is raised.
Yeah, really nice talk. I was just wondering if you could look towards the future and think about what's the impact of new technologies are going to be in this realm. Do you think?
Well, I think in terms of allowing, I work with a lot of breeders that are, I would call them on a human scale, meaning it's, one breeder and their team. And I think that it's been challenging for these individual groups to migrate into the realm of genomics-informed breeding because of the various constraints in terms of what it means in terms of reorganization of how the work is done and the cost of accessing this technology. In the big multinational corporations, they have highly integrated teams that can carry out all of this work on a very, very large scale. And we just haven't been as successful, I think, in doing this in the, let's say, public or smaller private breeding realm. And I think that by developing technologies that are suited to their needs in terms of, they can afford the cost of migrating into this, I think it's a really good opening to start. I haven't considered or given any sort of indication of how we're using these technologies more broadly in the lab. I focus really on the technical aspects, but I think another area where there's a lot of potential is in the realm of genomic resources or gene banks. And so typically, you can now interrogate these very large collections of lines, generate very high density catalogs of SNPs. And once you've done that, you can do a much more informed management of these resources. And so we've argued, for example, that these resources can be used to exploit genetic resources in a more precise fashion. For example, if you're interested in disease resistance, you can characterize SNP haplotypes around loci that are known to control a particular disease resistance and structure your gene bank and say, well, these 50 accessions have the same haplotype, therefore they likely carry exactly the same resistance, whereas these other ones have something different. If you didn't have that knowledge, you might need to analyze each resistance source individually in a QTL study, or you'd be going blind and making crosses, assuming, well, if I'm varying the the source of resistance, I may be differing the actual resistance gene itself. And in fact, many times they're shared. And so by having this extra information on what the underlying cause may be, I think it's a very, very helpful guide in terms of making decisions. And then just the basic management of, any gene bank is going to be full of either errors in identification or duplicates or all these other situations that are bound to occur. And by having access to very affordable genotyping technologies on a broad scale, it really enables to enrich the information that's available to gene bank managers to help do a better job of managing and better exploiting the resources that are there.
Oh, okay, thanks. I think that's, I suppose a follow-on question from that is how transferable are the data sets between runs, and how much effort is needed in sort of databasing those results to make that happen, I guess.
So in terms of overlap, we typically find that when you're operating with the same service provider that is using the same protocol for the same species, the overlap is actually quite high, meaning above 60% for sure of the loci being captured in different panels. Part of that discrepancy is simply because, some SNPs are non-informative in some panels versus others. So there's, in my view, a fairly high degree of overlap. And the other aspect is that certainly in barley, the LD is fairly extensive. And so data imputation or missing data imputation is quite effective. And so I think that once you have a good command of your basic germplasm and have high quality data for those sets, then doing any sort of imputation on sub-panels, if you will, or other sets of material is quite readily done. Now, obviously, in terms of keeping this information, as with any sort of large-scale information collection process, it does require some aspect of data management and housing these data and being able to share them, definitely.
Yeah, thanks.
Hello. Yeah, myself, Dr. Sorry.
No, just go. Yeah.
Yeah. I have one question, sir. This is a wonderful presentation, but in silico digestion, you have mentioned you can optimize the digestion of the fragment of that one. Could be used for the other crop also. You have done on soybean. If I have in rice system, I can use same platform for that one for optimization.
This depth finder tool can be used in any species. So basically all you have to do is provide your reference genome. So it informs the software on the sequence of your genome, and then it can simulate in silico digestion with whatever list of enzymes or enzyme combinations you provide it with. The one caveat that we have in plant systems that is a bit inescapable is that high levels of methylation can exist in some regions of the genome, and there's no way of knowing a priori if a site is methylated or not. And so obviously there's always some empirical testing on the side that's required. So the in silico predictions cannot take into account methylation levels that you cannot know beforehand, but they provide a good guide in terms of what could be a promising set of enzymes. And so for us, certainly they become a good tool for guiding our choices. And I didn't detail it, but for the low density GBS protocol in barley that I described, we did run some simulations to see if the SBF1 MSP1 combination would be a promising solution for that particular application.
Okay. And sir, did you come across in rice which enzyme is more like more compatible to the rice? Did you have work on some collaborative work in the rice in GBS?
If there's something that you would like to collaborate on, we can always talk it over, send emails, and examine that and see what can be done.
Okay, sure. Thanks.
There is a question. Is there any ways to tweak the reference genome to increase number of meaningful SNPs?
I guess what this might allude to is if you had, obviously, when you're working off a single reference genome, it always provides an incomplete picture of what the source of variation could be within a species. And so if you were able to run these tools off a pan-genome data set or reference genomes where you had a more complete picture of DNA sequence variation within barley, that would be one way to increase the number of SNPs. But I guess in our case, even if you design your protocol based on an incomplete picture provided by a single reference genome, whenever you conduct the analysis, you will capture SNPs that are the result of variation that may not be reflected in the reference genome that was used to design the panel. I guess it's a question also what reference genome you're using when you're doing the bioinformatics aspect of things. But that's one way in which I think in future there will be efforts to use more broadly representative sets of data as reference genomes and not a single reference genome using, for example, graph-based representations, graph genomes to more fully capture variation.
I have a following question for you about this. Now the published pen genome in barley is about 20 genome. The 10 are working to increase this diversity panel to, I think, 70. Do you think that it will be enough for you to map your genotyping by sequencing data analysis with this set of reference?
It always depends on what is your purpose. So when people are expanding reference genomes, some groups, I know, for example, in the soybean world, they've included many glycine sojas, which are wild relatives of soybean. And by doing this, you dramatically increase the number of polymorphisms that exist within what you're calling soybean. But if you're somebody who's working with domesticated soybean glycine max, those SNPs are not really relevant to you because you're working within the domesticated side. So going back to the barley story you mentioned, it just depends on how broadly representative these accessions are relative to what somebody is doing. So if you're doing very broad germplasm characterization in barley as a species, including land races and all these sorts of things, a panel of that dimension might still be insufficient. But if you're working within the realm of domesticated barley, especially elite material being used in very practical-minded breeding programs, then I would probably think that that panel would be very, not fully exhaustive, but provide a very good characterization of the type of diversity that's encountered in much more practically minded work, I would suspect.
How can you conciliate with structural variation data set? Can you incorporate this data by adding more reference? Challenging.
Yeah, so capturing structural variation is still very difficult. It requires resorting to mostly long read sequencing. And it turns out that even the analytical aspect of interpreting long reads to identify at least some types of structural variants are very difficult. What we found recently looked at comparing SNP-based GWAS, structural variant-based GWAS, and K-mer-based GWAS. And what we found was that using K-mers was a nice way of being capable of capturing signatures of all types of variation that might exist within a collection of germplasm. And it did a really good job of circumventing the need to do the characterization of structural variants, which is a fairly demanding task. And so if I were to give advice to somebody who wants to do, for example, GWAS and is concerned that by using only SNPs, that they might be missing some important determinants of this or that trait, I would tend to maybe recommend exploring K-mer-based approaches because they would simultaneously capture both the SNP derived or cases where SNPs are causal and cases where structural variants are causal. It's still quite demanding in analytical skills, but in terms of feasibility, it I think is a really nice approach for doing this.
I think it was probably unclear. Can you use cheaper by sequencing compared to resequencing in reference genome and kind of impute or predict some based on this growing assembly resource the genotyping by sequence?
We have looked at using GBS as an indirect means of calling structural variants, but I don't think it would be a very efficient means offhand because the majority of structural variants in plant genomes are going to be small insertions and deletions. And because of their small size, the chance of capturing them through direct GBS is limited. Where you can use the GBS is through a haplotype-based approach, where basically you have SNPs that will be in high LD with a structural variant, and they indirectly inform you on the presence or absence of a given structural variant. So that certainly, when considering very large numbers of accessions that where you would like to build on high quality reference genomes, but that obviously capture a limited number of individuals and you want to expand that, certainly I think that something like a high density GBS or skim-seq is a nice way of capturing enough marker information that you can then assign specific SNP haplotypes as being associated with underlying structural variants that may have been uncovered through broader, deeper sequencing efforts.
Okay, thank you. Maybe we can finish with the last question in the chat box. Would it be possible to preset select genome as a guideline for basis to help in synthesize bacteria construction?
I'm a bit at a loss here in terms. I'm not sure synthesizing bacteria. I'm assuming synthesizing bacterial genomes, but this is way off. I'm not into synthetic biology, and I have very little knowledge of how you go about designing or synthesizing a complete genome. So I'm afraid I don't really have an answer for this.
Thank you today for your disponibility. Very nice talk. People enjoy. There is a lot of questions. Was very interesting. Thank you. Thank you very much.
You're welcome.